the Audiovisual Studies Center, who we thank for their support. This forum has been possible thanks for different people that have supported us. The Health Sciences School, the UABC, the United States-Mexico Border Health Commission, the Binational Border Health Thematic Network, as well as the National Center for Preventive Measures and Disease Control, and the National Network for Research for Control and Prevention for Tuberculosis in Mexico. And this forum is taking place during the month of border health in the US and Mexico. It is important to point out that the purpose of this forum is to promote collaboration to prevent, diagnose, and treat tuberculosis TB under the new scenario with COVID-19 in order to be prepared for possible further pandemic. And since we're talking about prevention, our colleague Jorge Ariapasa from Peru, president of the Civil Association toward the life of those affected, has asked us to share this message with you today.
Este es un mensaje ideal para todo el Perú y el mundo. De Asisabi a Perú. Jorge Ariapasa. Con flema por 15 días, acude ya, acércate al centro de salud. Si tú tienes fiebre, estás muy flaco y no comes, acude ya, acércate al centro de salud. Si los doctores te dijeron que tienes el mal de tuberculosis, no tengas pena, no tengas vergüenza de lo que pasó, porque eso son cosas de la vida y tiene cura y es gratuito. Un saludo para el Centro de Salud Jaime Subieta y la doctora Yané Manta Ángeles.
Muchísimas gracias a nuestro colega de Perú. Creemos que
Thank you so much to our colleague from Peru. We believe that any means that we may use to strengthen prevention action is important, and a good way to start the day is starting it with music and, of course, with an excellent message. I would like to remind to follow that have been following our sessions. We remind them that we will give through the QR code, you will be able to receive certificates that you have taken this, that you have been a part of this forum. If you are a part of at least 6 sessions. Now, today we have 3 speakers. First of all, Dr. Francisco González and Sofía Lozano Díaz, a medicine student from the Mexican Institute of the Social Security and from the Monterrey University, and a doctor from Preventive Medicine, Health Public, and Microbiology from the Autonomous University in Spain. We would also like to ask our speakers to avoid speaking too fast so that our interpreters, our sign interpreters, as well as our English Spanish interpreters can get your message and can interpret adequately. We also remind each one of our presenters that they have 45 minutes for their talk, and at the end, we will leave us a Q&A space for all of our participants that follow us through the different social media. So now we're going to ask Dr. Gonzalez as well as Sofía Lozano to begin presenting their work title Latent Tuberculosis in Migrant Travelers to the Northeast region of Mexico, and we'll be taking your time, and we'll let you know when you have 5 minutes left on your presentation. Thank you so much. And please take it away, Dr. Francisco and Sofia. Thank you for being here with us. Good morning, everyone. I am going to share my screen. I hope that
ahí está, gracias. Doctor.
Muchas gracias. Bien, esta es una
okay, this is a presentational,
en la que
perdón, this is a presentation which we put together with my student,
Sofía Teresa,
Sofia
para que
Teresa,
presente
and she's going to present to us
este trabajo que hemos publicado
the work that we
recientemente.
have recently
Yo creo
published.
que
And I think
estoy sembrando una semilla
that I am planting a seed so that the commitment
para tratar de detener la tuberculosis, sus complicaciones,
to
sus efectos,
prevent TB, its complications, as well as its effects, and
con esta
to
enfermedad
promote
se
having
mantenga
more
y
people committed
durante más
to fighting this
generaciones.
disease
Eh,
throughout
Mañana amanecí un
future
poquito
generations. This morning,
con la garganta un poquito inflamada y
I
amanecí
have a sore
sin nada
throat, and
de COVID, andaba un poquito ocupado en la mañana,
I have just done my test for COVID, so I apologize if my voice cannot be heard perfectly.
la invitación
First and foremost, I would like to thank the organizers of this second International Virtual Forum for TB 2021 for the invitation. This is a very important forum at an international level,
es un evento
and it is an event
honra que me hayan invitado. Es
for which I am honored to have been invited. It is a privilege being here today with all of you, and I'm going to take advantage of these to share some of our experiences with you in the northeast regions of Mexico.
tuberculosis latente.
As far as latent tuberculosis,
Año pasado
last year
publicamos
we
este trabajo,
published this
colaborando
work
con
to
investigadores de la Universidad de Monterrey y
after having worked together with people from the Monterrey University as well as
estuvimos
with people
recolectando
from the
2 o 3 años
IMSS or the
en República
Social Security Mexican Institution.
de México. Y bueno,
And
el tema, vamos a estar hablando en las próximas
we're
sesiones
going
muy
to be
probablemente
talking about
acerca de la tuberculosis
latent tuberculosis infection in the
muchos de
next
nosotros conocemos
2 sessions. And a lot of
lo
us
que es la
know
tuberculosis
or are
latente
familiar with
y
what
vamos a estar
latent tuberculosis infection
solamente
is.
señalar
And I would like
algunos
to
de los
point out
aspectos epidemiológicos
some of the
de
epidemiological
esta condición
aspects
de la
of
tuberculosis
this
latente. Aprovechando
condition.
de que hay en el auditorio personas
Because I know that we have in our audience today people that might not be
relacionadas con este término.
completely familiarized with this term. When we talk about tuberculosis, we're talking about an infection, an active infection where the patient starts suffering from coughs, fever, weight loss, and they start feeling bad in general. But this condition is known as active tuberculosis. But when we talk about latent tuberculosis infection, we're talking about, well, in countries where there's a great prevalence of tuberculosis, this tuberculosis infection occurs. And a lot of us who live in countries with high prevalence had this tuberculosis infection during our childhood. We developed the symptoms, and our body and its mechanisms contained the infection, but it is never completely contained because this keeps living in our body in an asymptomatic way because it still lives in our body. But after that period when we had fever, cough, during our childhood, the infection was resolved, and then we never get any other symptoms. We simply have it in our body, but without any symptoms. And this condition occurs during our childhood, during childhood phases in most countries with a high tuberculosis prevalence, and it can prevail for many, many years. So a person can live with this latent tuberculosis infection, and you could even die with it, and nothing is going to happen to you. Sometime during our lifetime, though, when our immune conditions are affected, our system can be compromised, and then these can start, these infection that we still have in our body might start presenting symptoms. And it is considered that at a world level, there's 1,000 millions of people with these latent tuberculosis infections. And this represents at a world level 25%, 25% of the global population. For Mexico, we do not have epidemiological data because we do not have epidemiological research in general population in order to determine the prevalence of latent tuberculosis infection, but through studies done in other countries, it is considered that about 24% of our population in Mexico suffers from latent tuberculosis infection. Now, this is a mirror as to how tuberculosis prevails in Mexico. We have an active prevalence of 14.6 cases per every 100,000 people. And the state of Nuevo León has a higher prevalence, estimated at 24.4 cases per 100,000 people, and the state of Coahuila has 13.8 cases per every 100,000 people. There are conditions under which people that have this latent tuberculosis infection can develop active TB, and the conditions are diseases that compromise the immune system. All diseases that it's part of all of the diseases that compromise the immune system, but especially right now, we understand that we have a lot of obesity and diabetes. And in many publications, it has been mentioned that immigrants are people that can develop active tuberculosis after having had the latent infection for many, many years. The immigrant process is a process under which people migrate under different circumstances. And when they do this under legal and programmed conditions, they are not very likely to compromise their immune system, definitely. But a lot of people are migrating, and they have been immigrants for many years, and they have been migrating from South America to Mexico and then from Mexico to the US. Now, these immigrants that are migrating in an irregular form, in other words, without a legal document, during their path, they experience very harsh conditions where they have to bear high temperatures with very little water and food, and they have to sometimes walk thousands and thousands of kilometers. This is not the case for everyone, but there are people that are able to travel under more comfortable situations. But there are many immigrants that have to face all of these harsh conditions, and they are exposed to very high and very low temperatures, which affect their immune systems, and they might develop active tuberculosis. On the other hand, we can see through these
sujetos
publications
como alguien que tiene
that
tuberculosis
they point
latente,
out
lo cual es
immigrants
completamente
as
falso,
um,
porque ellos si tienen
risk
la
factors that
condición de
can
tuberculosis latente
spread
en el momento exactamente
tuberculosis,
en el
which
que
is
tienen
not
el
really
síntoma,
true.
no hay
If
ningún
they
problema con
suffer
eso, no se
from
dañan.
latent tuberculosis
Nosotros
infection, they cannot
queremos aclarar
spread this infection to anyone. And we would like for everyone to be clear about this because there are some work about immigrants in the US on active tuberculosis as well as on latent tuberculosis infection, but there's no evidence from Central America before that document anything that goes on with these immigrants before they cross the border to Mexico or from Mexico to the US. And with the objective of evaluating the immigrant population that have been traveling through South America, Mexico, and the US, using the which is the one that we use the most and which is authorized to diagnose latent tuberculosis infection in most of the countries around the world and of course in the US, which is the quantiferon TB Col Plus assay. I'm going to let Sofía speak a little bit about the different methods and proceedings that we used in order to complete this research work. So, Sofía, please take it away. Thank you, Dr. Well, now we're going to present the research methodology to you as a descriptive observational design was made at different shelters in Northeast in Mexico, where people that were identified as immigrants were given the test to identify latent tuberculosis infection. We also made a survey to find, could you go back, please? Okay, well, this was in general. Now I'm going to talk about population. As I had already mentioned, our population was an immigrants that were residing while in transit in two large shelters within the states of Nuevo León and Coahuila since January 2017 to October 2019. This study or this research study included 455 people over 12 years of age, and most of the immigrants were migrating to the United States from Central America or Mexico. But who were people who were excluded as part of this investigation? Well, people with chronic coughs and or with active TB were excluded, as well as positive smear and or positive cultures. People who were unable to respond to the survey and people who did not complete the questionnaire or whom blood test was not done. Well, after these exclusions, Doctor, could you go back to the last one? Okay. They all completed a survey. However, we were only able to collect 316 blood samples. Now, for the cross analysis, tables only include data from 297 people. Next slide, please. Now, as far as the design of the study, this was an observational, descriptive, and cross-sectional study. And this study was registered under by the Monterrey University Research Committee, and two shelters were reviewed. One of them is located in Nuevo, León, and the other one is located in Coahuila, and we paid um, visits regularly. Those who agreed to participate provided written informed consent. Written informed consent was also obtained from parents of participants for the people under no, in the age group between 12 and 17 years of age. And after the people were invited to be a part of this research study, and we took on the tests. Next slide, please. A standardized questionnaire was administered by trained public health personnel. Some of the information that we asked for was sociodemographic data, as well as medical history, including age, gender, marital status, country of origin, status, whether traveling alone or accompanied by others, health, and comorbidities, such as hypertension and diabetes. Now, as far as the procedures, after they had completed the questionnaire or the survey, the patients were given a somatometric evaluation, which included weight and height measurements used to calculate the body mass index. They were also given the option of blood collection in order to evaluate their glucose. And then after giving consent, blood samples were taken. For those participants who had their blood drawn, they were asked to fast for 8 hours prior to the blood collection. And these blood collections were divided into 4 milliliters of blood to fill the quantiferon tubes. The first one was done without antigens, and the tubes number 2 and 3 had TB2 and TB3 peptide antigens, which are the ones that are related to active cases of tuberculosis. And there was a fourth one that had positive mitogen. And these tubes were kept at 37 degrees Celsius, and the interferon gamma was determined as a response to collection tubes were evaluated by ELISA plaque produced in response to the TB antigens. Finally, the results were interpreted as positive, negative, or non-determined, and this was established by each software used. Now, as far as the statistical analysis, well, all of the questionnaires were kept in a database. The statistical analysis was performed using the statistical package of SPSS version 25.0. 0. And as far as the statistic analysis, the proportions well calculated, dividing the obtained volume from the total participants who answered the questionnaire. On the other hand, for the cross analysis, the data of immigrants from Costa Rica, Cuba, and Ecuador were excluded because of the insufficient data obtained. Now, the differences on frequency were evaluated using the chi-square test with a Yates correction, and the statistical significance was defined through another test. Now, the statistical analysis was defined as p minus or equal 0.05. What does this mean? This means that values of p under 0.05 were statistically important. Now, next slide, please. We're going to talk about the results. Next, please. Well, in this chart, we can see the country of origin for the different immigrants. In total, we evaluated 455 people. Most of them came from Honduras with a 71.1%. From El Salvador, we had 5.9%, from Mexico, 6.4%, and 4.6% did not answer this question. Next, please. Now, it is important to mention that the average age was between 29 and 10 years of age. The weight between 80. Social demographic characteristics of immigrants in shelters participating in evaluation for like tuberculosis infection. These are the data for Coahuila and Nuevo León. It is important to mention that 85.5% of our participants were males, 63.8% were single, and 49.8% had only elementary school studies. It is important to also mention that 5.8% of our participants suffer from diabetes, 26.5% were overweight, and 10.8% suffered obesity. The next one, well, what's important about this data? What we have found in our cross analysis was that patients from Honduras had a positive quantiferin of only 13.9%. And we have to remember that the Honduras patients were most of our participants in this study. But our participants from Nicaragua had 43.8% and El Salvador 41.7%. What does this mean? Well, this means that there's an important difference in the positive quantiferin according to the country of origin, finding a higher rate over 3 times higher in El Salvador and Nicaragua compared to Honduras and Guatemala. It is important to also mention Mexico, which was at 30.0%, which also means that in comparison to Honduras and Guatemala was a lot higher. Well, this chart can also show gender. There's not a big difference between males and females. 19.1% were males and 19.4 were females. Now, as far as marital status, there were no important differences. Next slide, please. And well, as far as education, no important differences were found. However, as far as obesity and as far as overweight and obesity characteristics, we found a difference where people suffering from overweight and obesity were 2 times more likely to have latent tuberculosis infection compared to those without these characteristics. And with this, I finish my presentation and I ask Dr. Francisco to take it away one more time. Well, um, some important things that we have mentioned about our results that for me were surprising and they were innovative because we found that as far as immigrants, most people were from Honduras and Guatemala. But the rates, the TB infectious rates among immigrants from these two countries, which represent 75% of immigrants that we interviewed, have a latent tuberculosis infection rate under what we have found in the immigrants in Mexico, as well as the ones from El Salvador and Nicaragua. Very likely, we are talking about rates between 30% for Mexicans and 42 or so percent from El Salvador and Nicaragua. Therefore, where the global rate for Latin tuberculosis infection is of 25% globally and 30% for Mexico, our results are congruent as far as the immigrants coming from Mexico and the global calculation.
But migrants from Guatemala and Honduras have a rate of tuberculosis that is very low. If we look at the active tuberculosis rates in the countries of origin, which is a mirror of these rates of latent tuberculosis, we see that in Nicaragua and El Salvador, they have rates of active tuberculosis. Currently in El Salvador, it's 70 per 100,000 inhabitants. But in the last 3 years in El Salvador have been awful in terms of the presence of tuberculosis. In previous statistics, it was about 42 or 43 at the levels where Nicaragua is at right now. In people with tuberculosis, with active tuberculosis per 100,000 inhabitants. In Guatemala and Honduras, we have about 32 and 28 per 100,000 inhabitants. So it's very likely that our results that we are seeing are the result of high rates of tuberculosis in the countries of origin. But if we compare this with our country, we see that 75% of migrants coming from other countries, other Central American countries, they have rates, they have far lower rates of latent tuberculosis than we have here in Mexico, and only 18%, which is for the migrants from Nicaragua and El Salvador, have higher rates than here in Mexico. And therefore, I'm inclined to think that these people who are migrating will exhibit a risk of developing tuberculosis due to the conditions in which they are traveling. But this risk is not greater than what Mexican migrants would have, and specifically people who are coming, people who are migrating from Nicaragua and El Salvador have a greater risk of developing tuberculosis during or after their trip to the United States than migrants from Honduras and Guatemala. Very good. On the average of latent tuberculosis in these migrants is 18.4%, which is much lower than the world average and much lower than the average in Mexico and much lower than migrants traveling from Mexico to the United States. So therefore, I do not agree with the idea of singling migrants out as presenting a risk of passing tuberculosis on. And on the contrary, it would seem that the environment on the border, the traveling and the conditions in which we are traveling could represent a risk for these migrants, a risk of that these migrants would develop or catch tuberculosis here on the border, on the Mexico-US border, or in the United States, in the border states of the United States where they have higher rates of tuberculosis than we found in this study. So in conclusion, there are places where the migrants come from, where they have high prevalence of active tuberculosis. So migrants from Nicaragua or El Salvador have a high rate of latent tuberculosis, and migrants from Guatemala and Honduras have low rates of latent tuberculosis. So controversially, in our data, we did not find any statistically significant difference, although there is a trend to latent tuberculosis for those who have diabetes compared to those who don't. But we did not find statistically significant differences. I think, and that's just a hypothesis, that the presence of diabetes in this relatively young population it's because they have diabetes, but they haven't had it for very long. My hypothesis is that when they've had diabetes for longer, the age is also a risk factor for latent tuberculosis. They will develop latent tuberculosis in a later stage of diabetes. We should separate people recently diagnosed with diabetes and those who have diabetes for longer. And Dr. Estrepo in previous presentations has noted the importance of diabetes in the development of tuberculosis infection, and we did not find that difference. We did not find any statistically significant difference, but we did find that in migrants with who are overweight and obese, they had a greater risk, and that was statistically significant. They had a greater risk of developing tuberculosis. Well, we would thank you for your attention. I am very grateful for the honor of presenting to you at this important forum today. I would like to thank the organizers of this symposium, this forum. Thank you very much. Thank you, Sofía, for all your support, because she has prepared practically all of the content of this of the presentation for this forum, and it was a great job. Thank you very much, and thank you to all of you. Thank you very much, Dr. Francisco González and Sofía Lozano. We would like to continue with some questions that we have received, and we would also like to invite the people who are following us on Facebook and YouTube. We would invite you to send your questions as well. So, first question is, based on your results, what are your recommendations as a research team for health authorities in Nuevo León and Coahuila in terms of the health care that should be given to migrants with latent tuberculosis? If we could make issue a recommendation for authorities, it would be to have migrant populations have an equal treatment, but migrant populations should have the right to healthcare and to epidemiological monitoring like anybody else in anywhere in the world. And for them not to be treated with discrimination, due to that these people should not be seen as a risk of carrying latent tuberculosis. Based on these results, that's not the case for most of them. We need to break this stigma, and if this research helps a little bit to achieve this aim, then that would be what I would want to recommend. Thank you, Francisco. Just a favor. Could you stop sharing the screen? Yes. Thank you very much. Another question. You say one of your exclusion criteria was that people had active tuberculosis. How many cases were excluded due to that reason? We did not find anybody with active tuberculosis. Okay, thank you. Very good, thank you. Another question related to the first one is, what are these strategies that the Health Secretariat has adopted both in Nuevo León and Coahuila to address and treat the health of the migrant population? Do they have any kind of program in particular, or how are they operating to care for these populations? In particular, it's we are not part of the Health Secretariat, and we are not familiar with the programs that they have. There is a commitment which is very significant by the National Commission, National Health Commission, Border Health Commission, and there are programs, and we are trying to achieve this equality in treatment and to get a dignified treatment for migrants. But I think that is insufficient. What we have seen in belonging to this commission, what we have shared with our colleagues who work in institutions in Tamaulipas, Nuevo León, and Baja California, and San Diego, Arizona, etc., is that there is a commitment to there's a commitment by health institutions to offer health services to these people, but I think that healthcare for migrants depends more on people's goodwill, and some people, rather than institutional processes about caring for migrants, I think we still have a lot to do to provide care as an institution. We are making progress, and I think we need to continue to create inclusion projects that we can implement that would include institutions. At this time, they are open to work together with us in preparing migrant care programs, but it's very institutional still. Thank you very much, Francisco. We have another question which says, has there been follow-up to people who tested positive for latent tuberculosis, and do you have any results from subsequent tests administered to these people? We have carried out several projects with the migrant population. The migrant population is a group that travels around a lot. They are here for 4 or 5 days, and then we don't see them again. But we have already done a follow-up study with these migrants, and I think we had about 200 in Arizona, which we did with the University of Arizona, and it was very difficult to locate them afterwards. Most commonly, they didn't answer our emails or messages or answer phone calls, so it's very difficult to follow up with them because these people are afraid of being discovered and being deported, especially when they're already in the United States and they've managed to get there. They don't want to be, they don't want to receive phone calls or emails or anything like that. It's difficult. Thank you, Francisco. Another 2 questions, and one which I think is quite quick. What is the average cost of a quantiferon test, like the one you used in the study? It depends. Commercially, it could be up to 1,000 pesos, but for us, 250 to 300 pesos. Thank you. The last question we have for you: did you include the variable of the incidence of tuberculosis of each country of origin in your statistical analysis? Okay, there are lots of questions in one here. Whether you combined overweight and obesity, which was about 33%, maybe that's why it was significant. They also asked about history of contact with tuberculosis. Yes, we did include obesity and overweight together. And yes, it was significant. The result was significant. The whole publication with the details that you're asking us for is with this title, with the same title as the presentation. It's online. It's open access. Thank you very much. About the question about contact with tuberculosis, did you not investigate that? We did ask, but there were no people who had been in contact with active tuberculosis. We didn't include it because nobody had that history. Okay, good. Thank you, Dr. Francisco González and Sofía Lozano, for your presentation and for answering these questions by the audience. Thank you. So we will listen to the presentation by Dr. María, which is latent tuberculosis, the success of living in secret. We remind you that you have 40 minutes for your presentation. And so when you have 5 minutes left, I will interrupt you to let you know. Okay? Thank you. Thank you. Good afternoon, good morning over there. First of all, I would like to thank those who have invited me to give this talk in this forum, which is so interesting and so necessary. And I'd like to tell you that I'm going to talk about the disease, or a latent infection rather, the people that are infected. I don't think that it's going to have the social connection of the last presentation had, which I congratulate you for. Tuberculosis is a complex disease, and it's defined as a spectrum. Can you see that? Can you see my screen? Yes, we see it. Yes. Okay. Tuberculosis is described as a spectrum of places without the disease and places with the active disease. We have latent tuberculosis. In latent tuberculosis, in general, we have it is characterized by the presence of granuloma, where we have bacteria in a latent state. The diagnosis of latent tuberculosis is with these tests which determine the response, the immune response by the individual against the tuberculosis infection. When we have these tests, these tests are not specific because they are positive in other conditions as well, not just latent tuberculosis. And it's important with respect to this situation, a situation where the infection has been eliminated, but the individual still has an immune response. There is no way of distinguishing it from the latent infection, because this is important, because these people who have an immune response but do not have the bacillum, they do not need preventive therapy. So it's important to know if the person really has a latent infection. It's not that easy. Recently, we have included another fifth step in the spectrum of infection, which is the incipient TB. This means that the bacillum is latent, and when it starts to awaken, because we say that it's dormant, and we say that it awakens, it's when that happens, it's incipient. The difference between these intermediate steps is mainly clinical, but what we do know, or what we do aim to describe, is that in incipient TB, there's usually fewer bacilla, so the bacilla load in the individual is lesser. What is the reason why tuberculosis is living with us and producing this latent state? We have evolved with tuberculosis. The tuberculosis has evolved together with us. I'm giving you these examples which show this co-evolution, this adaptation of the bacillum to our body. It has all these products which interfere with cell functions like phagocytosis, autophagy, and when we know how this works, so these products can co-activate this process or inhibit it based on what they need to survive. When the tuberculosis enters the macrophages and microcytes, it acquires a permissive situation. The cytophages are not able often to eliminate this bacillum, so it has to adapt to living within our cells. Latent tuberculosis was considered in the WHO strategy in 2015, which is called MTB. It was considered very important to control it, to know how much latent tuberculosis there is, to control latent tuberculosis and control active tuberculosis, which is important, which is what causes death. The definition of latent tuberculosis is written here, and is an immune response to tuberculosis antigens in the individual, which is defined as latent. One aspect is that they had this in the strategy, is that many of the cases of tuberculosis in developed countries are due to reactivation of this latent tuberculosis, and this strategy has produced quite good results. This is the WHO report this year. 30 deaths have been reduced by 35% and incidence by 20%. Therefore, it seems that we are doing well. The diagnosis of latent tuberculosis is performed by more modern procedures, by the IGRA and with tuberculin, and we have a third of the population is estimated to be infected with latent tuberculosis. But more recently, we have described, based on these studies, on various studies on annual risk of infection by tuberculosis in all these years, it is not a third, but it's a bit less, about a quarter of the population. Although the numbers are probably more accurate, but there are billions of people who are infected. So that means that over the course of these people's lives, there will be about 100 million people who will develop active tuberculosis. And so detecting it accurately is very important. But the problem with these processes is that they do not distinguish between a latent infection and a maintained immune memory, because what they are measuring is the immune response by individuals. So based on that, we believe that a positive immune response means that they have viable bacilli that are stimulating our immune system, and that's why we detect with these procedures this immune response. But the reality is not totally, it's not exactly like that. In this work, where we try to find out the proportion of reactivation in tuberculosis immunoreactive people, we want to know which proportion reactivated the tuberculosis, and we have found very, very low levels, only between 1 and 11%, depending on the study. Had reactivation of tuberculosis. What the studies say is that a large proportion of these people who are positive by these procedures may not carry viable bacilli, which means that they will never reactivate the tuberculosis. One study last year studied the transcriptional profile of TB positive and TB negative immunoreactive people. And what they found was that a fraction of people who are positive, the IgG positive, were clustered with they were clustered with IgG negative people, so as if they were the same thing. This concludes, they conclude that a proportion of these IgG positive people do not carry bacilli and will never develop disease because they are positive due to the immune response, not because they actually have the infection at that time. And that's very important that we do not, that we're not taking into account in this kind of diagnosis. And there's no other kind of diagnosis for latent tuberculosis. These people that we are detecting as positive are not carrying viable bacilli. The problem that we have with this procedure, what do we mean by having a bacterium that is dead? It's not that easy to define. At what point can the bacterium no longer resuscitate? According to this review, there is a point of no return. When the bacterium arrives at that point of no return, it's dead. But anything that is above that point means that the bacterium is alive. And one thing that we accept that determines whether a bacterium is alive is whether it has a metabolic activity. Is it functioning? Does it have proteins that are doing something? Therefore, we need to look at the metabolic activity to determine whether the bacteria are alive or dead. If a bacterium is dead or alive, one thing we can do to know whether it grows in a culture. So there are various commercial procedures that are used to determine if we have viable cells in a sample, and they are all based on whether the bacterium is whole. If it's not broken, we have different colorants. If it's green, it means it's alive. If it's red, it means it's dead. There is another procedure, but it's no longer with the microscope. It's with a PCR to determine whether the bacteria is okay. What we want to know, whether they are alive or dead with an intercalating agent, which is inhibited. If it's altered here, it's not integral, and that means that the PCR that is performed later will be negative. But if this procedure, if it's positive, it means that in that sample there are viable bacteria. What is the problem with these procedures? We know that dormant bacilli have have modified their cell envelope, and so they are not stained with colorants. So we need to look for other procedures that are not based on the cell envelope to determine if the bacteria are viable or if they are alive or dead. In this laboratory, we are finishing describing the study. This is not a study to determine if the bacteria are alive or dead. And what we've done in this study is to see in what phase on the tuberculosis spectrum can each of these approaches be useful. So we're going to see if we can have a procedure that will be useful, for example, in an incipient tuberculosis or a latent tuberculosis. We cannot apply all of these procedures. We have to see whether they work or not. But some of them, we have used them. The first two ones here, the ones we have used in our laboratory. And the first one is useful for all phases of the disease. But once we know, and we have these tools to know whether the bacteria are alive or dead, what we need here is to look in the granuloma to know whether the bacteria are alive. It's in the granuloma that we'll find them. These macrophages, foamy macrophages. This is not completely true because of various studies that have been carried out in the 19th century, when we found that some of the tissues that were normal from individuals dying from causes other than tuberculosis was inoculated into laboratory animals. These animals developed tuberculosis. So these tissues that were normal microscopically, there were viable bacteria in the donors to immunosuppressed recipients, where there were some tissues and some organs that were that later developed tuberculosis. In the spectrum, in where we can try to look for latent bacillum for tuberculosis, we were not able to publish this. We sent it to various journals, but none of them accepted it because maybe we didn't explain it well. This was in collaboration with a doctor from Mexico, and and we found that there was DNA in tuberculosis without granulomas in adipocytes and macrophages, to see if that was related to tuberculosis or not, but to other bacteria as well. We used another microbacterium because of the we only found in the macrophages. So that means that we were not able to publish it, but a few years later, the publication, Dr. Hernandez Pando, we found DNA in bone marrow human adipose. So that was similar to what we had found, and maybe we didn't. They didn't like how we explained it. The relation between tuberculosis and adipose tissue is quite clear, not just because of what I've just told you, but what I'm going to tell you in a minute. And for example, in mice that were infected with tuberculosis, this comes together in the tissue. What they did was, with a chemical, they decreased the amount of adipose tissue. The bacilli migrated, and then in this case, the mouse had the infection, tuberculosis infection, we must not lose sight of the fact that one of the most typical characteristics of tuberculosis is consumption, which is a weakening of the individual weakness. So the fact that tuberculosis ate away at the adipose tissue has created a pro-inflammatory environment, which, when sustained, can lead to the death of the adipocytes. We have also used a similar situation. In this study, what we did was to use stem cells, hematopoietic stem cells, which are long-term and pluripotent. We have these this acronym here. And what the authors say is that it gets into these stem cells, and from there it is maintained, and then they develop the disease. This happens without going through the granuloma. And what we have to remember also is that some time ago, Robert Koch suggested that the latent infection in extrapulmonary tissue, first we have the extrapulmonary tissue. So where do we look for these viable silent bacilli? We can look for them in many niches in myeloid cells, and then these different types of cells. And one thing that I want to talk about adipocytes, the reason why tuberculosis likes being there in the adipocytes is that the duration is much longer, so it's like a more stable house. Because when cells divide or die, they have to migrate to other cells. What these authors suggest in their hypothesis is that tuberculosis penetrates into the stem cells, and then a pluripotent cells, and then they go here in. And recently, this year, we described this work where the bone marrow, where there's an alteration in the balance between myeloid lineage and lymphoid lineage. That's a characteristic that tells us that soon there will be clinical manifestations of tuberculosis. That means that when the homeostasis is broken between these two lineages, that means that announces to us that the tuberculosis process is beginning. So far, we have an idea about where tuberculosis might be hidden. But one thing that we found in the laboratory, maybe if we find where it is, maybe we can develop processes to eliminate it. And now I have to talk about the similarity between these two concepts, which are persistent and dormant. This is very similar, where we have a reduced division and low metabolism, and there are situations that are often confused. But persistence and latency are very different things. But persistency, in persistency, the bacillum is alert and aware of the changes, but it comes close to stress. But in latency, that's what we want to find. And here, it is maintained like this until there's a drastic change and it has to awaken. So we have studied the genes that are associated with persistent bacteria and those associated with dormant bacteria. In the case of persistent genes, the genes that are most associated with the situation are toxin and antitoxin systems. These are systems with two components that have all the bacteria, and in the case of dormancy, we have the genes that are associated with tuberculosis are those of the lipid metabolism, taking into account what lipids like, what this bacteria likes. The tuberculosis has a lot of systems, and it has a lot of systems, and these systems have a very curious characteristic. These antitoxin systems are leaderless. They do not have promoters. They have a site where they begin to synthesize to create the proteins that are going to create the antitoxin system. So these leaderless, these are archaic promoters. These are less effective than modern promoters. So the hypothesis that is suggested here is that the bacteria has evolved from an environmental bacillum to a human or mammal bacillum by optimizing the adaptation to its host, by maintaining the characteristics of a persister and acquiring the capabilities of the dormant one. The first study where the lipid association was described, which is a very important gene, so that the lipids, so that the bacteria can eat the lipids. And we saw that the mice that were infected with this mutant did not develop the disease. And this was the way that the researchers were told that this was important in the latent infection. This table shows what the bacteria likes. If we have the oleate medium, and this the macrophages accumulate as lipid drops, which are these green drops here, but then this is eliminated. But what happens if we also have these? But here we have the ability to keep this in the environment that it likes. And then after the 2 days, the macrophages are still full. So what do we do to study the latent bacilla? What we have to do, we are working in latent models, is to imitate what happens in the granuloma. These are the conditions that normally occur in the granuloma: hypoxia, nutrient starvation, acid pH, nitric oxide. There are many models of in vitro dormancy. We have a model of hypoxia. We know that hypoxia does not respond to all the problems, all the questions. But what we have done is use medium and even LCFA. I'm going to talk a little bit about that. Using these in vitro procedures, we have a latent phenotype. This phenotype is the loss of resistance, and a typical form of addressing tuberculosis. The drug tolerance to antibiotics and slowing of the division. What we suggested was to use a medium which was rich, so we gave the bacteria something to eat. We chose to use an even number of carbon atoms, because this produces acetyl, which is not toxic. But if we have an odd number, we have a toxic product, which is 3 carbons. 3 atoms. So this was research what we did in collaboration with a group by Dr. Gonzalez from Mexico and Dr. Portillo from Colombia. And what we did here was to compare the different phases of growth and the exponential and stationary phase, what happens to the bacteria if we grow it with a dextrose medium or an LCFA medium as the only source of carbon. And this is where the different bacteria in these different conditions. The first thing what we saw in the stationary phase of growth in the fatty acids, we have an accumulation of droplets which did not appear in other conditions, and this also generated antibiotic tolerance. This means that in our conditions, in a stationary phase, in a long chain fatty acids, but what we found was very interesting, because what happened was in these stationary phases, what happened in the stationary phase was that the amount of transcribed RNA molecules used by the bacteria, which was codified for protein, which was very high compared to those mapped to CDS. When we started to analyze which ones they were, we found that many of them were cationic amino acids. The tRNAs were molecules used for the synthesis of cells, and here there was no significant synthesis of cells. So what we suggested was to use the modified cell envelope, which means that the antibiotics cannot break through it. So the other ones were the non-coding RNAs. There are many of them. Some of them are studied, some of them have not been studied, and they are participating in regulatory processes that often are unknown. This is the panorama of the bacteria when they grow. And we have the stress, it has a reductive stress, and to eliminate this stress, which accumulates lipid droplets, this protein synthesis is blocked in the envelope here, which is what makes it tolerant to antibiotics. And the non-coding RNAs is what we want to look at in more depth, to see what is happening, to see what function these non-coding RNAs have. But one factor that we need to look at in this study is hypoxia, which is related to the establishment of latency. When we have a normal level of oxygen, the number of mycolic acids is greater than when the bacterium is latent. And when it is reaerated, which is when we reintroduce oxygen into the culture, which was in a state of hypoxia, the number and the amount of mycolic acids come back to normal levels. So what we proposed in this project was to introduce into the hypoxic study into what we were analyzing. This is the hypoxic model. He described 2 phases of hypoxia. One phase where the hypoxia is softer, it's just 1% oxygen, and another phase where it reduces more than 10 times. It's reduced by more than 10 times, which is just 0.06 100% oxygen. So what happens when it goes from the exponential phase to this first phase and then to the second phase, if we have a source, a carbon source or lipids, I'm just going to give you some results. One of them concerns the non-coding RNAs. This concerns the non-coding RNAs in the hypoxia with the carbon source, and where we have the same ones, where the long chain fatty acids. Most of them are small non-coding RNAs. It slows down the growth of the bacteria, and this occurs both with dextrose and long chain fatty acids. Some of them are in the three conditions. This is a bacterium survival molecule, which is a signal recognition particle. This is an RNA molecule, which is when the protein is being synthesized, and it takes this protein to the site of the cytoplasm where it is functionally valid, which means that it's helping the bacteria to transport the proteins to the site where they are needed. And then we have the tmRNA. What this does is when the bacteria is in a difficult situation, they get stuck and they do not finish producing the proteins they need. So what this does is it unblocks them and eliminates this and releases ribosomes so that the protein can be synthesized. In where we are working with data from hypoxia, we have this work in which Dr. Gonzalez is also from Mexico is also working, and in this work, what we did was use the same system, but by adding cholesterol, which is a very abundant molecule in our bodies that the bacteria can use. And we have hypoxia, but only in the first phase. So I'm going to show you some comparisons of the data from this work where we have the cholesterol and our data without cholesterol. One of the comparisons is about the transcriptional regulators. When we have this in hypoxia, we have this central regulator which appears, and there are many central regulators, up to 37, which are overexpressed in hypoxia. But when we have the fatty acids with a carbon source, there are just 2. This is what we're saying. The bacterium does not need to use a lot of these. So what they do is manufacture or create more, because these are numbers that maybe are not that interesting to you, but I just wanted to say that when we add cholesterol, the number of transcriptional regulators increases drastically. And the toxin antitoxin systems in our situation is lower. And I'm just going to skip over this and just say that dyslipidemia, so an alteration in the levels of cholesterol, makes the bacterium be more susceptible to tuberculosis infection. This is the conclusion. And the bacterium is in hypoxia in a steady state response, and we have the transcriptor, but here with the cholesterol, we have a stress response, which means that the bacteria, when it is in a lipid state, hypoxia is not really an important factor. I just wanted to show you the project that we have started now, which is to add this mix of lipids and iron to the abundance of iron. We have the different phases. We have one phase which is on the right-hand side here, and another phase here on the left, which is in the laboratory. Because of the hospitals, because of the pandemic, we haven't been able to start, but we are starting now in the study of tuberculosis at the transcriptional level and in vitro. We have a lack of iron. And now I'm not going to bother you with data about this, but we're going to have a series of different culture media in different conditions, different growth conditions, and each of these conditions will be studied with and without iron. So I hope to be able to talk about that in the future. This is just the final message, a final takeaway message. This latency is the result of a host pathogen adaptation. And this latent tuberculosis could be in tissues that are not do not have granulomas. And what is clear is that the presence of lipids is a determining factor for the bacteria to develop. So what we need to know is, because of these studies are in vitro approximations, which are very different to the reality of the patients. I would just like to thank all of you we have worked with, and this is the team that we have in the university, and here we have these students, and I would like to thank you. Thank you for listening. I have finished. Thank you very much. Thank you. Okay, thank you very much, Doctor. There's just 1 minute left. I would like to thank you very much for your presence. Not all of the audience who are with me, we are not all experts in this topic, so it was a very illustrative presentation, and has given us a very good idea of all the future work. And of course, we hope that by next year you have results from your study. We would love for you to tell us about it. And we have a few questions for you. The first one is there a difference in the tuberculin presence between the different types of tuberculosis, the latent and active tuberculosis, etc.? Sorry, I don't know. I didn't understand the last part of the question. I will repeat the whole question. Is there any difference in the tuberculin test and the different types of tuberculosis, the latent or incipient tuberculosis, etc.? We haven't seen that difference. What has been studied, especially, we use a tuberculin test with the interferon gamma. And what has been described is this difference between latent and subclinic, etc. We haven't seen, for example, that in the latent there is a greater response. It's just goes over the threshold, and that's how we determine whether it's positive or not. What we have seen is that the difference in the different phases has been seen from a clinical point of view. So it would be interesting to see that from the point of view of the markers from the patients. So there must surely somebody who is studying that. So we could see markers from the patients, which would give us information at the level of the patient to know whether it wouldn't tell us whether the bacteria is alive or dead, but that would be very interesting. One question that we have here for you, which says, Could you tell us, could you explain briefly the difference between a persister and dormant bacteria and how this is reflected in the clinical presentation? In the clinical presentation, there is no way of differentiating them. Let's just say the persister bacteria is in a situation where there is something that is bothering it. For example, the classic persistence is the presence of antibiotics. When you expose the bacteria to antibiotics, they can survive in such a way that they do not grow in the culture, but they remain viable. And these bacteria are called persister or persistent. In latency, dormancy, they have tolerance, and it remains. The bacteria remain latent whether or not the antibiotics are there. The difference is that the persister bacteria must have something that is bothering it at that time, so that it will survive. To make it try to survive. So the key factor is the environment. There is a hostile environment in the case of the persister bacteria, and with the latent bacteria, it is not a hostile environment. In the case of tuberculosis, we put them into an environment with lipids, and the bacteria remains there. The bacteria remain there, but without creating its own elements that help it to survive, because it's not in a hostile environment. Maybe that's clearer, easier to understand difference. With regard to your research and your development, what is the participation of the iron in the normal growth? In the latent, is that a factor for growth? That's what we are aiming to study. What is the role of the iron in the establishment of the dormancy? Iron is one of the basic metals for any living being to be alive. And so it helps with many functions of our body, and it helps with many functions of the bacteria. So what we want to do is put the bacteria into a medium and to see the difference if there is or isn't iron. So we see if the dormancy is established or not. But so far, the participation of the role of the iron has just been part of a study where it's been described in a list. I don't know, maybe it had too much data. But the in vitro dormancy, one of them is the lack of iron. Well, we know that when the bacteria is in the granuloma, it has to go through a lot of things, and it can't get to the center of the granuloma. So during dormancy, the lack of iron is standard. What we don't know is, or the only thing we have studied is to grow the bacteria. But this was a standard model. This is the iron that is used in a minimum medium. And what we've seen is that the bacterium stopped growing. When the bacteria stop growing, it is always assumed that they enter into a state of dormancy. What these authors did not study is that the only marker of tuberculosis, of latent tuberculosis, what they studied was that it was tolerant of a bacteria with little iron and with antibiotics, but they did not see an accumulation of lipids or the other characteristics. So that is what we are aiming to look
Excelente, muchísimas gracias. Entonces, definitivamente sí, el próximo año.
at in more depth. Thank you very much. Thank you very much. We will hope to see some more results from you. Another question: in a context of growing bacterial resistance to antibiotics and high prevalence of obesity in populations, what do you expect to be the scenario in terms of tuberculosis? Okay, that's a question, but maybe I'm not the best person to answer it. The results that we are seeing, that we are trying to show you in the presentation, is that the presence of lipids is good for establishing a latent infection. So what they are telling us is that the obesity is a disease in itself. In obesity, we have an inflammatory process that alters what I'm going to say. But if there is an accumulation of lipids, this prevents the latent bacillus from resuscitating. So that's why it's important to make sure people are well fed and end the state of famine that exists in many parts of the world. Because obesity is an disease and is a comorbidity, an pre-existing condition for other diseases. This is not bad with reference to tuberculosis. As we were saying, when the adipose tissue is reduced, that's when we can enter into an active state of active tuberculosis. One last question. In your opinion, how far are we from using a marker with a transcriptional base to differentiate them quickly and in an affordable, accessible way? We are not far from that. It depends what the marker is, how you want it. If you want it from the individual, from the infected person, I think there are many studies that would be the subject of another talk that would determine that there are certain markers that tell us that the individual has latent tuberculosis or is going from a latent infection to active tuberculosis. The problem, this is with regard to the individual. With respect to the bacterium, we need to know whether it's alive. Once we know it's alive, we can describe it. We can say that the individual has a latent infection. What happens is, talking about something that I'm not an expert in now. The grading, the degree between the different types of tuberculosis has a clinical basis, and this is often subjective. So it's often not easy to know if individuals that have certain symptoms, but these are not. So we classify that as a subclinical or as an incipient infection. I'll leave that to the clinicians who are experts, and they know how to classify this. But we need to make sure that's clear. The individual who has an incipient tuberculosis, we are taking him where the bacillum load is not that high. So at that time, it would be good to define, to stop the disease. But maybe when it's at a later stage, we cannot control it as easily. Thank you. I think the information that you're giving us is extremely valuable. Okay. Thank you for your attention. I'm sorry, I had a noise here, so I muted the microphone so that it wouldn't be recorded. Okay, so we have no more questions. Thank you very much, Dr. Garcia. We know that for you it's almost time to go to bed. So Thank you for this information. Thank you, Dr. Francisco Gonzalez and his student whose name escapes me. Thank you for your presence, for your presentations. I would just like to thank you. Thank the Mexican Sign Language interpretation team and the language interpretations team into Spanish, and of course thank you. Thank SEPA for the support in broadcasting the session through the social media and through YouTube. We will see you next week. Next week we have our fourth session of the forum, where we will have Dr. Roberto Cuevas from the Public Health Institute in the University of Veracruz in Mexico, and Dr. Lozano from the Tec de Monterrey. So thank you very much. We will see you at the next section. Before I forget, remember to record your attendance, otherwise you won't be able to receive your certificate of attendance. Thank you very much. Thank you and have an excellent day. Thank you very much. See you soon. Bye-bye. Thank you. See you soon. Bye-bye.